we have discussed the cell division, the interphase, the mitosis. In interphase we discussed and detailed the G1 phase. In G1 phase, the genes, the proteins, the pathways, GQ pathway, GS pathway, RAS-RAP pathway, and state pathway, all are discussed. But before we really go to DNA replication, we have a need of crystal concept of DNA. What is DNA? What is the structure of DNA? Right? What are the components of DNA? What is nucleotide? What is nucleoside? What are the functions of nucleosides and nucleotide beside the structure of DNA? The functions of nucleotides other than DNA structure. Right? So, before DNA replication, we will go through DNA structure and function. But before DNA structure and function, we will give just a look to the location of DNA and cell. Where is DNA? Where is the house of DNA and cell? Right? So, in structure of cell, we have discussed the nucleus. Nucleus. What is nucleus? Nucleus is a central part of cell. This is the central part of cell. This is the capital of cell. Right? Like all the activities of our government in Pakistan are controlled from Islamabad, which is the capital. Like that, all the activities in cell are controlled by nucleus, which is the capital of cell, which is the central part of cell. And why nucleus is responsible for this controlling? Because DNA is present in nucleus. Right? So in nucleus, we have discussed that nucleus is composed of three parts. It is made up of three parts. The first one is nuclear envelope. Nuclear envelope. The second is nucleoplasm. And the third one is nucleolus. Nucleolus. Right? Nuclear envelope is composed of three main things. One is outer nuclear membrane. What is the main characteristics of outer nuclear membrane? Ribosomes are attached to outer nuclear membrane, and ribosomes are responsible for protein synthesis. So, if we have nucleus here, for example, this is nucleus, right? So, this is the outer nuclear membrane, right? And tiny particles are attached to this outer nuclear membrane, which are called ribosomes. And ribosomes are very important in transcription and translation, right? After outer nuclear membrane, we have another membrane here called inner nuclear membrane. So the second thing is inner nuclear membrane. Inner nuclear membrane, right? What is the main characteristic of inner nuclear membrane? Lamins proteins are attached to this membrane. Lamins proteins. Lamins proteins are responsible for attachment of, right? What is the main role of this lamins proteins? It attach the heterochromatins or prochromatins, right? And prochromatins, right? And if mutations take place in these lamins protein, progeria will cause, and progeria is the quick aging of a baby, right? So these are Lamins proteins. These black are Lamins proteins, right? And this membrane is inner nuclear membrane, right? The one important thing here, the pores are present, the nuclear pores. What is the structural component of these pores? Anyone? I am not asking about the function, the structural component. Proteins. These are made up of proteins and these are extrinsic proteins, right? So these are nuclear pores. What is the main function of nuclear pore? Exchange, transportation. Nuclear pores, right? All the things which wants to leave this nucleus will follow this nuclear pore, and all the things which are needed inside the nucleus will follow this nuclear pore, right? Collectively, these three things form nuclear envelope, right? The next thing, this nucleoplasm. Nucleoplasm is the soluble part, or the part where the semi-solid liquids are present inside nucleus, inside nuclear membrane, right? So this portion is called nucleoplasm. And nucleoplasm is very important because it holds the chromatin. It holds chromatins, the thing which we need today, chromatin, right? But I have told you that chromatin is present in two forms. Euchromatins and prochromatins are heterochromatins, right? These are also chromatins, right? So chromatin is present in two forms. One is pro or heterochromatins, and the second is euchromatins. From this E, you can remember this expression, genes expression. The chromatin which involved in genes expression is euchromatin, right? This prochromatin is tightly packed with Lamins proteins or inner nuclear membrane. So these are attached with inner nuclear membrane, and euchromatin present in loose form in nucleoplasm, and these are the chromatins which are responsible for genes expression, right? And the chromatin is made up of DNA plus histone proteins, and today we will go in a very big detail for DNA and histone proteins, right? The next thing is nucleoplasm, because nucleolus is very important because DNA is responsible for the storage of heredity material or the genes or the storage of all the information of our body. And what is that? So chromatin is composed of DNA and histone protein. But here is a very important structure present in nucleoplasm, which is called nucleolus. The nucleolus is the nucleus of nucleus. It is the nucleus of nucleus. And this nucleolus is responsible for the attachment of two things. One is ribosomal RNA and the second one is proteins, and forms ribosomes. Ribosomal RNA plus a specific type of proteins formed It is mean that it forms the small units and large units both of ribosomes, right? But fortunately, ribosomes are very tiny and can pass through these nuclear pores and comes to cytoplasm. Attached to outer nuclear membrane or endoplasmic reticulum are also present in the free form of cytoplasmic ribosomes, right? So this DNA is responsible for forming three things. One is ribosomal RNA, messenger RNA and transfer RNA, right? And this is the reason why we call the DNA as the storage information in our body, right? Because we study this DNA in which field? Genetics. The field which is for the study of DNA is genetics, the study of genes, the study of DNA, right? So we can say that DNA stores all the information about the structure and function of an organism, right? And every part of the organism, right? How? Because this DNA is responsible for making RNA, right? This RNA maybe this is ribosomal RNA, maybe this is messenger RNA, and maybe it is transfer RNA, and these all the RNAs are responsible for protein synthesis, right? This messenger RNA is responsible for taking the message from DNA, ribosomal RNA is responsible for reading that message, and transfer RNA is searching for the anticodons of messenger RNA, right? And transfer that amino acids and making proteins. And which type of protein it will make? Which type of proteins it will make? This is the thing which we differentiate the humans from other animals and other animals from other animals, right? Gore, the ribosomal RNA, messenger RNA or transfer RNA for protein synthesis the parada responsible or specific type of proteins जोड़े दादन सही रहा नो उस दा कम type of proteins जोड़े कम type of सीजन नोड चे कम responsible ली पर cell की दन the function the parada कई नो दा reason चले दा सा कई चे the whole organism दा यो बल न जुदा जुदा की is responsible for making RNA, and this RNA is responsible for making proteins. These proteins may be structural and functional proteins. Structural proteins and functional proteins. Right? The structural proteins are the proteins which form the hairs, right? The hairs, the bones, etc. Nails. The functional proteins are hormones, enzymes, etc. Right? They are following the different pathways in our body. And these structural and functional proteins determines the species. Either this is human, either this is cat, either monkey, right? So for this reason we can say that DNA stores the information, all the information, right? So we will dive now in the structure of DNA. Chromatin is formed of, broadly it is formed of chromosome. If we condense this chromatin, we will see chromosomes, right? And in every species there are a specific number of chromosomes. In humans it is 46. Mean in every cell, if the chromosome number are 46, this cell will be of humans, This is the cell of human, right? So we will take one chromosome from these 46 and we'll zoom it and we'll see that how this chromosome is made up of DNA and how this DNA is formed, right? So we will take one chromosome here. For example, this is our centromere, right? And we have this chromosome, right? What is the tip of this chromosome called? Telomere. Telomere. Why telomere is important? Anyone? Why we separate this part of chromosome? Because here the tip of DNA is present, and from the tip, for example, we have DNA, and DNA is always double stranded structure, right? One end will be 5 end and second is 3 end. This is telomere, right? On one side phosphate group will present, and on other side OH group will be present, right? So this is telomere. This red is centromere, right? And these are sister chromatids. If we want to know more about chromosome have sister chromatids, right? This short chromatid is called p-arm and this long is called q-arm, right? Kulo ko to दादा रात को टाइम दे, right? This is q arm and this is p arm, right? But this is one end DNA or two end DNA. This is 2 end DNA having four sister chromatids. This is one end DNA, right? So after replication, the chromosome will become like this, right? Now we will take, this is the strand of DNA, right? Chromosome is made up of two things. For example, this is chromosome. What are the things? What are the components? DNA plus histone proteins. Histone proteins are specific for DNA, and right, how many types of histone proteins are present here? 2 types. We will discuss in just a few seconds, right? We will zoom this chromosome. When we zoom this chromosome, we will see that this chromosome is made up of fibers. For example, this is the fiber out from this chromosome, right? And these are called the loopy continuous fibers, right? This is the loopy continuous fibers because continuously it is looping. Looping mean das is tavigi convolutions dipadeki, right? So these convoluted fibers are called loopy continuous fibers. But when we zoom these loopy continuous fibers, we will see that these fibers are tightly packed. Like this, right? And these fibers are called These are called, anyone? These fibers are called highly packed helical fibers, right? Highly packed helical fibers, right? And these fibers are around a small proteins, right? For example, these are that proteins, right? These are looping these proteins, and these proteins are called histone proteins, right? So now we will zoom these loops, right, and we'll see that how it made, right? For example, we have zoomed these proteins. These are histone proteins, and around these proteins the DNA is coiling like this. For example, right, the DNA is coiling around these histone proteins. On each histone protein it forms two loops, right? Mean DNA two time looped around histone protein, right? So these red are histone proteins, and this green is DNA. This green is DNA, for example. But there is another protein present here, right, which connects these units, right? For example, this thing is this is also histone protein, and this is also histone proteins, but there is some compositional difference between these two histone proteins. Both are histone proteins, right? We will take one histone protein from here and we'll zoom that histone protein, that what is the difference between this blue histone protein and this red histone protein. So for example, we take this histone protein here. This is the red histone protein. This red histone protein, around which DNA make two loops, right, is composed of 8 units, 8 other units. This is histone protein, and this histone protein is composed of 8 units. That's why this is called octamer, having 8 units. So we have 8 units here. For example, 1 2 3 4 1 2 3 4 5 6 7 8 units, right? And what are the names of these 8 units? Anyone? One is H2A, second is H2B, third is H3, and 4 is H4, right? And every unit is present 2 times, mean 2 units are present of each histone protein. So 2 units are H2A, 2 units are H2B, 2 units are H3, and 2 units are H4. The total is 8 units. So it is called octamer. If someone asking from you that what is histone protein, what are the types of histone protein? There are 2 types. The one is around which the DNA is looping, and the second one is which connects that loops, right? This histone protein is also known as octamer, which is composed of 8 further units. One is this blue histone protein. This blue histone protein, right? This histone protein is called H1 histone protein. This is also known as octamer, and this protein is also known as, anyone? It is also known as linker protein. Linker protein, because it links the two units of DNA, right? So this is some overview of these loops. After this, we will zoom this green fiber, which is DNA. Histone protein is finished. There are two types of histone protein. The one is H1 and the second one is octamer. Octamer is made up of 8 further units, H2A, H2B, H3, H4, right? And this histone is H1 histone proteins, right? What is the composition of this histone protein? Anyone? This is protein, so it will be made of amino acids. The amino acids are lysine and arginine, right? And both are positively charged amino acid. So mean that this histone protein have positive charged. It have positive charge, and after some time we will know that DNA have negative charge. For example, this is DNA, and DNA have negative charge. Because of phosphate group, right? So negative and positive attracts each other. Negative and positive charges attracts each other. So for example, this is histone protein and having positively charged, and this green is DNA and it have negatively charged. So these both will attract each other, right? So by this charge attraction, the histone protein and DNA makes bonds. They are together, right? If we add some positively charged or some negatively charged to this DNA, it will affect the structure. It will affect this structure, and we use that differences in the structure due to charges and many treatments of DNA, right? If we want to lose the DNA, we can. If we want to tight the DNA, we can, right, by these charges. So this is the bond between the histone protein and DNA, right? The histone protein माने कम यो charge दे positive, पर DNA माने कम यो charge दे negative. That one charge is able attract कई. No jidai attract कई, no the wajina the histone protein or the DNA चिरन the able sarasari tightly packed. पर ये attractions मानदे. No kamang ugwalu namanga da luskulium shuli kalil tamang nor positively charge or ponda ba attraction noram. O kamang negatively charge worku the had na sewa na sabak igda buluskigi. Daga dadai namanga faida alu or faida sara jamung ba treatments kida sizune use ko. Ta so gore pa drugs bane charges dikana. Your drug ke chana negatively charged debal positively charged de acidic de yo alkaline de ole the DNA structure chede da acids o heat o pade vanem effect kigi. Manga da uncoiled cooling shoe manga da denature cooling shoe har sa cooling shoe ठीक ना. So this is the story how the DNA and histone proteins are together by attraction of negative and positive charges, right? Now we will zoom this green fiber which is DNA. So this is right, this is double helical structure. The double helical structure of DNA was discovered by Right? This is the structure of DNA, the general structure, having two strands. And these two strands, well, one will always come from parent, father, and the other is from mother, right? So this is the double helical structure. But before we go to the structure of this nucleotides and nucleosides, just look, this DNA have grooves. One is this small groove, right? This, and one is this large groove, for example this one. So if we talk about the grooves of DNA, we have two grooves. Group is like that. DNA have grooves. One is major groove and one is minor groove. Grooves of DNA. Grooves of DNA. One is major groove and the other is minor groove. Right? What is the importance of this groove? The gene's expression, the DNA will uncoil from this major groove. Right? The space between one groove and other groove, one small groove and other small groove, is called nucleosome. Right? For example, if we take the distance from here, this is minor groove, this is minor groove, and this is also minor groove. So the distance between one minor groove and other minor groove is nucleosome, which is composed of mainly 10 nucleotides. 10 nucleotides. And we can stop the DNA replication by tightening this minor groove. If we, for example, if we give something and by that we hold this, for example, this is, it holds this minor groove. After holding, the enzymes will not able to uncoil this point and DNA replication will be prohibited, inhibited, right? So we can treat cancer by this way, by holding this minor groove, right? So this is nucleosome, this is major groove and this is minor groove, this is minor groove and this is major groove, right? This is nucleosome, the distance between one minor groove and other minor groove. This is nucleosome, right? For example, if we take this distance, if we take this distance, this is one nucleosome and this one nucleosome is composed of 10 nucleotides. This is your paper MCQ, right? But remember one point here, this 10 nucleotides are only specified for B form DNA. B form DNA is very abundantly present, right? This 10 nucleotides, if the nucleotides become 11 or 12, it will form A form DNA, and if these nucleotides between one minor groove and other minor groove exceed the 12, it will form Z form DNA, right? So this is for only B form DNA, and last we will discuss A form, B form and Z form of DNA in detail, right? So this is the nucleosome. Now nucleosome is composed of nucleotides, right? And nucleotide is composed of nucleosides, and nucleoside is composed of other components, ribose and nitrogenous base. Now we will really discuss this structure because these are the nitrogenous these are the nucleotides. For example, this is adenine, this is thymine, this is guanine, cytosine, adenine. It will make bond with thymine, adenine, cytosine, guanine, thymine, right? We will zoom now this area and we'll see that what are these things, right? This is called nucleotide. If we take the sequence, right? First we have phosphate plus nitrogenous base plus pentose sugar. It forms, if we add, right, this nitrogenous base and pentose will form nucleoside. When we add phosphate here, it will form nucleotide, and if we take a lot of nucleotide, it will form nucleic acid. But here we are discussing DNA, right? So how we can define DNA? The DNA is the sequence of nucleotide which are formed of nucleoside and phosphate, right? For example, we have a lot of nucleotide sequence, and this nucleotide is composed of nucleoside and phosphate, and or simply we can define that the DNA is the linear sequence or the linear polymer of nucleotide, right? This is the DNA is the linear polymer of small units called nucleotide. So now if we learn the structure of nucleotide, we will easily learn the structure of DNA, right? First we will learn the general structure. After that we will go for the chemical structure slowly, right? For example, this nucleotide is composed of three things, right? One is nitrogenous base, right? The second thing is pentose sugar. The third thing is phosphate group, right? After learning this DNA structure, we will compare this DNA with cell membrane, right? And you people will ask me the differences and similarities, right? So one is phosphate, the second one is nitrogenous base, and the third one is pentose sugar. Collectively these three things form nucleotide, and nucleotide forms DNA, right? What is nitrogenous base? How we can define nitrogenous base? There are two types of nitrogens, purines and pyrimidines. But what is nitrogenous base? These are the heterocyclic nitrogen containing aromatic rings, right? These nitrogenous bases are heterocyclic nitrogen containing aromatic rings. One ring or two ring form this nitrogenous base. So on the base of these rings we divide this nitrogenous base. One is purines and pyrimidines. Right? Purines have two rings. Always purine have two rings. Generally we can say that this is one ring, and this is other ring, having 2 rings, right? And pyrimidine have 1 ring. And last we will take the chemical structure of these rings, right? On carbon number 7, carbon number 1, carbon number 3 and carbon number 9, these carbons are replaced by nitrogens in aromatic rings, and that's why these rings are called heterocyclic, because it contain the things other than carbons, right? And they are called nitrogenous base because they have nitrogens, right? So this pyrimidine is made of 1 heterocyclic aromatic ring, and this purine is made up of 2 heterocyclic aromatic rings, right? Purines are adenine and guanine, adenine and guanine, right? And these pyrimidines are cytosine, uracil and thymine, right? These have 1 aromatic ring and they have 2 aromatic rings. In DNA, adenine, guanine, cytosine and thymines, these nitrogenous bases are present in DNA, right? And adenine, guanine, cytosine and uracil, these are present in RNA, right? The only difference between on the basis of nitrogenous bases, the only difference between the RNA and DNA is this nitrogenous base. RNA have uracil and DNA have thymine, because in RNA thymine is replaced by uracil, right? So these are nitrogenous bases, we will discuss in detail, right? The second thing is pentose sugar. Why these are called pentose? Because they have 5 carbons. These are also rings, aromatic rings, but are homocyclic, having 5 carbons, right? So pentose sugars are of 2 types. One is ribose and deoxyribose. Deoxyribose. In DNA, deoxyribose is present. Deoxyribose is present. For example, we take this pentose sugar here. For example, I have this 5 carbon ring. Carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. And this is another pentose sugar, carbon 1, 2, 3, 4 and 5, right? What is the actual difference between ribose and deoxyribose? Here on carbon number 2, OH is present in ribose, right? In deoxyribose on carbon number 2, 1 oxygen less than this ribose. 1 oxygen is less than ribose, so it is called deoxyribose, right? This is, and this deoxy is present in DNA and this ribose is present in RNA. Ribonucleic acids and deoxyribonucleic acid, right? So these are the pentose sugars which are present in nucleic acids, right, and nucleotides. The next thing is phosphoric acid. Phosphate, and this phosphate is responsible for the making the backbone of DNA, the backbones, right, which are the strands. The strand is connected by phosphate group, right? So we will discuss this in detail now, but the composition of nucleotide is phosphate, nitrogenous base and pentose sugar. Not all the nitrogenous bases will present in one nucleotide. Only one nitrogenous base will present in nitrogen, and these nucleotides play a lot of roles in our body. beside the DNA structure. It have a lot of functions also, right? So we will discuss this in detail after a short break, right? Sok ne apoi no mata uai. Take the DNA. DNA, we have discussed this chromosome, and human have 46 chromosome. Each chromosome is made up of DNA and histone proteins, right? Tell the name of these fibers. Loopy continuous fibers. Tell the name of these. Highly packed helical fibers, and these helical fibers are looping around these proteins called histone proteins, right? This red is histone protein, and this histone protein is also known as octamer because it is composed of 8 units. That 8 units are H2A, H2B, H3, H4. Good. 2 units, 2, 2 and 2. These are full 8 units, right? One other histone protein is this blue. This is H1 histone, also known as linker histone proteins, right? After that, we have zoomed this DNA. DNA is double helical structure. What does DNA stands for? Deoxyribonucleic acids, right? This is DNA, and DNA is composed of nucleotides, right? So now we will discuss this nucleotide, right? The sequence of composition of DNA is nucleosides plus phosphate forms nucleotide, and these nucleotide forms nucleic acids. The nucleic acids are DNA or RNA, right? So we can define DNA as this is the sequence of nucleotides which is formed of nucleoside and phosphate, and nucleoside is composed of nitrogenous base and pentose sugar. So we will go now for the structure of this nucleotide. What is nucleotide? Nucleotide is the building block of, this is the building block of nucleic acids. Nucleic acids mean DNA or RNA, right? So if like nephrons, from nephrons kidney is formed, from neurons brain is formed, and from glucose carbohydrates are formed, from amino acids proteins are formed, from fatty acids lipids are formed. So if we know the structure of these units, this is the structure of the whole polymer. Like that, if we have the knowledge about nucleotide, this is the structure of DNA, right? So nucleotide is formed of 3 things. Nucleotide have 3 components, right? The one is nitrogenous base. For example, this is nucleotide. Nucleotide is composed of 3 units or 3 structural components make one nucleotide, right? The first one is ribose sugar, pentose sugar. The second thing is nitrogenous base. The third is phosphate group. Right? These three things collectively form nucleotide. Right? The first one is pentose sugar. Pentose sugar is 5 carbon sugar unit aromatic compound. Right? Each carbon in this ring have their own importance. Right? But we have two types of pentose sugars. The one is ribose and second one is deoxyribose. Right? This ribose sugar is present in the nucleic acid RNA and this deoxy is present in DNA. Right? What is the difference between this ribose and deoxyribose? Let's take at its structures. Right? For example, we have 5 carbon ring. Right? This is carbon number 1, carbon number 2, this is carbon number 3 and this is carbon number 4 and this carbon 4 is attached with carbon number 5. Right? To this carbon number 1 nitrogenous base will be attached. Nitrogenous base. This is the importance of each carbon in pentose sugar ring. Right? This carbon number 2 will differentiate the RNA from DNA. How? In DNA and RNA, this carbon number 2 have hydroxyl group, 1 oxygen and 1 hydrogen. Right? What is the importance of this carbon number 3? That it will form the diester bond with phosphate group, the next pentose sugar. Right? This carbon 4 is common and this carbon 5 is attached to phosphate group. On one side it will attach with 5 number carbon, on another side it will attach with this carbon number 3. Right? But the actual difference between ribose and deoxyribose is this carbon number 2. This carbon number 2, because in ribose on carbon number 2 OH group is attached, present. Right? Now take the deoxyribose. Deoxyribose. Right? In deoxyribose, why it is called deoxy? Having 1 oxygen less than ribose. Right? This is carbon number 1, this is carbon number 2, this is 3, 4 and 5. Right? Like that, carbon number 1 is attached with nitrogenous base, carbon 3 will be with phosphate, carbon 5 will be with phosphate. This is phosphodiester bond. It will form the phosphodiester bond. It is called diester because it will attach 2 times. Right? Phosphate. And this carbon number 2 differentiate this deoxyribose from ribose because this carbon 2 have only hydrogen. Right? In deoxyribose, carbon 2 is attached with hydrogen, and ribose, carbon 2 is attached with hydroxyl group. This is the important, this is the difference. Right? The next thing, the nitrogenous base. There are 2 types of nitrogenous bases present: purines and pyrimidines. Right? What is nitrogenous base? What is nitrogenous base? Heterocyclic aromatic compounds are the aromatic rings which is composed of nitrogens, right? Having nitrogens are main component. Purine have two aromatic rings and pyrimidine have one aromatic ring. The general structure, remember this general structure, right? It have one 6 carbon ring and one 5 carbon, right? Remember this forever. In this we will count the carbons anti-clockwise and furines anti-clockwise. For example, this is the first nitrogen position, this is the second nitrogen position, this is the third nitrogen position, this is the fourth nitrogen position, right? So this is 1 2 3 4 5 6 7 8 9, right? This is anti-clockwise counting, right? In pyrimidines, one ring, one benzene ring is aromatic ring is present of 6 carbons compounds, right? Here the nitrogen positions are right, but here we will count clockwise 1 2 3 4 5 6. This is clockwise counting. This is internationally accepted methods of counting the carbon. In furines we will count anti-clockwise. Do you know what is anti-clockwise? What is anti-clockwise? How ulta? The watch ulta. The watch ulta. They the anti-clockwise way. Adal tabung the watch sarap parallel chalu. Take the here in pyrimidines we will count the carbons clockwise. Clockwise, right? So the nitrogenous bases in which two rings are present are adenine and guanine, right? The nitrogenous bases having one carbon ring is thymine, cytosine and uracil, right? But remember that adenine will always bond with, it will always make bond with thymine, right? Which bond? Double hydrogen bond, right? And guanine will always, for example this is guanine, it will always make bond with cytosine but triple hydrogen bonding. So which one is very strong among these? Guanine and cytosine, right? This is also the importance of this bonding, that we can denature this DNA here easily and here not easily, right? So this is the bonding and these are the ring nitrogenous base. But now what is nucleoside? What is nucleoside? If we attach the nitrogenous base with ribose or deoxyribose, right, pentose sugar, it will form nucleoside. Nucleoside. Here phosphate is not present. Phosphate is now not present yet, right? For example, if we take adenine, this is adenine, nitrogen and nitrogen, right? here it has right. This is for example adenine, right, and we bound it with which thing? Ribose. This is ribose sugar, right? So it will form nucleoside, and for example these two rings are of adenine, and this red is pentose sugar. These both collectively form nucleoside, right? After this we will attach the phosphate group to this nucleoside and it will form nucleotide, right? But there is some nomenclature for these nucleosides, right? When these nucleosides are formed, for example, if we take adenine and pentose sugar, it is called adenosine, right? If we take this guanine and pentose sugar, it will form guanosine, right? If we take thymine and pentose sugar, thymidine, it will form thymidine. If we take uracil plus pentose sugar, it will form uridine. If we take cytosine plus phosphate, it will form cytidine, right? So one accepted and very easy nomenclature there is that in pyrimidines with nucleoside we will add iodine, right? And in purines osine will add it to the name. So adenosine, guanosine, thymidine, uridine and cytidine, right? Understand? After this we have phosphate group, and phosphate is negatively charged, and due to this negatively charged the whole DNA holding negatively charged, right? This is phosphate group, right? It will form the phosphodiester bond, phosphodiester linkage with pentose sugar, and this is one of the strongest bond in DNA, right? If we compare this hydrogen bonding with connects adenine with thymine and cytosine with guanine, and the other bond is 5 to 3 phosphodiester linkage, right? This is the strongest, this is a strong bond, and this is weak, right? For breaking this phosphodiester or for breaking this hydrogen bonding, less energy is needed, and enzyme can easily break this bond. But for breaking phosphodiester bond, we will need nucleases. Right? Nucleases may be DNases and RNase. We will need these, right? And some of these are exonucleases and some are endonucleases. For example, we have DNA. For example, this is DNA, and some enzymes came and break this from middles in both sides. These nucleases are called endonucleases, right? And if enzymes came, nucleases, and break this DNA from sides, these are called exonucleases. nuclease, right? So collectively this phosphate, nitrogenous base and pentose sugar will form one nucleotide, right? Generally, if for example this is the nucleoside, for example this is the nucleoside, right? And this is the pentose, this is for example phosphate group, this is phosphate group, right? And this thing is nitrogenous base, this is nitrogenous base, this is pentose sugar, and this is phosphate group, right? Collectively it will form nucleotide, right? So when we talk generally about the DNA, we will make nucleotide like this, and after this we will make it chemically, generally but chemically, right? We will form the rings, right? So for example the sequence of DNA will be like this. For example, we have sugars, and these sugars are bonded by phosphate, right? And these sugars are attached to nitrogenous base, adenine, thymine, cytosine, etc., right? This is one strand DNA, right? So now we will discuss the structure of DNA after this. Now I have told you that this is the nucleotide, which has three parts. One is pentose sugar, nitrogenous base, and phosphate group, right? And DNA is the sequence of nucleotide. So we take the sequence of this nucleotide, right? For example, this is sugar. Which sugar? Deoxyribose or ribose sugar. And DNA, actually deoxyribose is present. Deoxyribose sugar. This is another deoxyribose. And this is deoxyribose, right? This is called the backbone of the strand of DNA, right? And this backbone is attached, for example, this is carbon number 5, 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 and this is carbon number 3, this is carbon number 3, this is also carbon number 3, and this is carbon 3. This sugar units are attached by phosphate molecule like this, right? And this is what? This is nucleoside or nucleotide? This is incomplete nucleotide because it's only deoxyribose, pentose sugar, and phosphate group. For completing, we have nitrogenous base. This is nitrogenous base, right? For example, this is adenine, this is guanine, this is thymine, and this is cytosine. This is also, for example, adenine, right? The next strand, this is one strand. The next strand is anti-parallel to this. The DNA strands are anti-parallel to each other, right? So the next strand will be opposite to this. For example, this is the nitrogenous bases. These are the nitrogenous bases. right? The anti-nitrogenous base to adenine is thymine, guanine, adenine, cytosine, and thymine, right? But this trend is opposite. For example, this is that person with showing nitrogenous bases, right? It will be, for example, this is deoxyribose sugars, deoxyribose sugars. This is deoxyribose, this is deoxyribose, this is deoxyribose, and this is also deoxyribose, this is also deoxyribose, right? And this backbone is attached by which thing? Phosphate. This is attached by phosphate, right? Now this is the head of this nucleotide, and these are the feeds of nucleotide. Mean that on this side 5 number carbon is present, here 3, here 5, here 3, here 3 number carbon, and here 5 carbon, right? And these are the hydrogen bonding between nitrogenous bases, right? So we say that this is anti-parallel to each other. The strand will always make from 5 to 3 end, 5 to 3 end in DNA. This is, for example, we say that this is the left side of DNA and this is the right side of DNA, right side, right? So for example, this is left side and this is right side. How these are anti-parallel? The left side strand is from 5 to 3 end, right? And this right side strand is 3 to 5 end. These are the complementary strands. Complementary because all the nitrogenous bases are anti of each other, and thymine is always making bond with adenine, right? And all the DNAs are right stranded except Z form. Z form is left handed and the A and B forms are right handed, right? For example, if I am a nucleotide and I have this is nitrogenous base, this is pentose sugar, and this is phosphate, right? So when I want to make bond with another nitrogenous base, it must be in opposite direction, right? Come here. For example, this is one nitrogenous base, right? Nucleotide, and this is another. This is the nitrogenous base, and this is nitrogenous base. Can these nitrogenous base can make bonds in this way? No. If this hand is able to make bond with that, right? For example, this, right? This is left stranded and this is right. Just like this, these strands are in anti-parallel form because the nitrogenous base is present on right side, and right cannot make bond. One must be in opposite direction. Like for example, this is that nucleotide and this is this. If I want to make bond, I will revert it, right? So this is the reason. Thank you. Right? So this is the structure of DNA, general structure. In S phase, we will go for the replication of this DNA, right? We will uncoil, we will unbond this nitrogenous bases by enzymes, specific enzymes, DNA polymerases, right? And will make other strands with these two strands, right? And replication, for example, if this is the DNA, this is the DNA, right? And I want to uncoil this, so it will become like this, right? And the nitrogenous bases will separate it, right? One strand will form with this strand, right? And one strand will form with this strand, right? And remember, I told you that in each 2N DNA, one strand is original and one is copied, right? These are now two DNAs. These are now two DNAs, but this is one strand is original and one strand is copied. In DNA replication, we will discuss this phenomena in detail, right? So this is the structure of DNA. Now, how we can stop DNA replication to diminish or finish the cancer, right? I have told you that DNA have two types of grooves, right? This is the minor groove, right? DNA is not just like this that we make on board. It is just like this, right? These are the nitrogenous bases bonding in DNA, right? So we have two grooves. One is minor groove and one is major groove, right? If we want to treat the cancer, we have to stop this cell from entering into S phase. For example, this is the interphase, this is the G1 phase, and this is S phase. This is the G1 is checkpoint, right? We are trying to stop this cell here to go into G0 phase, right? So by stopping the DNA replication, we can achieve this, right? How we can stop this DNA replication? We have to stop this uncoiling of DNA. If we stop the uncoiling of DNA, we can easily stop the DNA replication, right? So we give blood drugs. What is this? This is duck. Right? We need a drug to hold this minor groove, and now the DNA polymerases are not able to uncoil this DNA, and this drug is called, is like duck, so it is called ductinomycin. Ductinomycin is anti-cancer drug, and it stops the DNA replication, right? So this is one way to avoid the cancer or to prevent the cell from cancer, right? The next thing, we can by acetylation reactions, we have one subject, this is genetics, and one other subject which is called epigenetics. Epigenetics, and epigenetics, for example, epigenetics, right? For example, I have DNA here, and this DNA is coiled around histones proteins. Epigenetics, we can if we modify DNA, it is called epigenetics. So we can modify the DNA and also we can modify the histone protein, right? For example, if we modify DNA, called the modification of DNA, right? For example, this is DNA. Learn this. This is important for you. Not this. This is the study of children, right? And this is the study of doctors, right? So if we modify this DNA by adding which thing? Methyl group. Where? To the major grooves. Methyl groups, but not too much. Only one methyl group will be attached to this, right? Or we add acetyl group, acetyl group. It will prevent this DNA from genes expression, mean from transcription. Transcription will be prohibited, and when transcription prohibited, the cell is preventing from cancer, because now the cell cannot make more proteins and will not goes to S phase, right? The next thing, if we want to modify the histone proteins, modify histone proteins, right? For example, we have DNA here and it is around histone proteins. If we add acetyl group to this DNA, acetyl to this point, right, the DNA will loosed like this, and now these genes can do genes expression because they are in loose form, right? So if we want to enhance the transcription, to increase the transcription, right, we can add acetyl group, right? Like this, if we add one methyl group, the same phenomena will take place and transcription will be enhanced. But if we add more than one methyl group, if we add more than one methyl group to this DNA, it will become more tight like this, right? And in this case we can prevent transcription. These are the general points about the treatment of cancers, right? After this, the DNA replication can be stopped by some drugs which we are using in routine drugs, right? The side effect of their drugs is DNA replication inhibition, right? Some of that are sulfonamides, antibiotics, right? This is a class of antibiotics. The hydrolazine, one of the safe drug in pregnancy, right? Isoniazid, anti-TB drug, right? The next is procainamide, and procainamide is anti-arrhythmic drug using for heart, right? This is routine drug. The next is penetrine, which is anticonvulsive, which is used against the fits, झटके, पर झटका नहीं पखल कुछ राजी convulsions fits, the treatment apara chikam drug worki penetrine. It also can stop the DNA replication, right? Some other drugs, furines and pyrimidines inhibitors, right? For example, anti-fungal drugs, which is used for fungal infections. For example, anti-cancer drugs. For example, antiviral drugs. It can also stop the DNA replication and can affect the body, right? So this is all about the structure of DNA. It is very simple, but these points are important for you people, and generally that is important, right? Understand? After this we will go for DNA replication. This is enough for today, I think.